Oh, maybe not yet.
I will make sure to edit out the introduction.
How you doing, James?
Right.
Thanks for doing this.
Yeah, happy to.
Yeah, no, I think it's great for people to get to hear about the exciting science that's happening and why they're signing people up for studies.
Great.
You're warming a wine collection in the sun there.
Yeah, old champagne bottles from like defenses and graduations and things like
I
that.
see. Okay,
Yeah,
all right.
that's right.
Okay.
Oh,
Just getting them in the light.
yeah, that's right. That's right.
Okay, it's a long weekend coming up, and I think we should get started. Thank you all for coming. Dr. Alvarez is here to talk to us about some very exciting research he does at the bench and in looking at a translational model in the mouse. Uh, and he trained at Harvard and also at the University of Pennsylvania for his postdoc, and has been out here looking at cell dormancy and resistance and residual disease, all the things that we worry about, and trying to figure out how we could look at this earlier to overcome treatment resistance. So thank you very much for joining us. And I'm really glad we can renew this series and keep going with these talks so that we all can know what everybody else is doing around here. Thank you, James.
Great. Thanks for the invitation and the introduction. Can folks hear me okay, I hope? Maybe someone can give me a thumbs up. Okay, good. Uh, Thank you. Okay. Yeah. So as Hannah said, I really am a cancer cell biologist. I want to start with that disclaimer because we think about things from the perspective of cancer cells and their signaling pathways and metabolism and so forth. And from a clinical perspective, we're really interested in how aspects of the biology of cancer cells affects things like drug resistance and tumor recurrence and so forth. But I really want to emphasize that my expertise is in sort of the cell biology of cancer. So I really appreciate this opportunity to talk to clinicians and folks who think about breast cancer from a clinical perspective, um, so that we can, try to make sure that some of the ways we're thinking about residual disease and what I'll call persister cells or tumor dormancy and eventual relapse are sort of clinically relevant. And I'd love to get feedback on ways that the audience thinks that, the models that we're using and the approaches we're taking either are or are not clinically relevant, and maybe how they could be improved and how some of the pathways that we're interested in could ultimately be translated. Um, so switch to a laser pointer here. So these types of slides, these sort of few intro slides are probably unnecessary for this audience, but the point that I'm trying to make here is that, ideally, following treatment of a primary tumor in many epithelial cancer types, including breast cancer, the goal is to eradicate all of the cancer cells in the body and achieve a cure. However, we know that in many cases that's not achieved, and there are residual cancer cells or a residual tumor, also goes by the name minimal residual disease, that is left over in the patient's body. And these can be at the primary site in the case of neoadjuvant therapy, or they can be disseminated throughout the body, can also be found in circulation in the form of circulating tumor cells. And I think there's circumstantial evidence that a fraction of patients that have residual cancer cells will actually never go on to develop a recurrent tumor. And so the residual disease in these patients would be stable and they would, for all intents and purposes, be cured in spite of residual tumor existing in their body. But the patients about whom we worry and the ones that are really motivating this are those in whom these residual cancer cells start to regrow and form a recurrent tumor. And again, this tumor relapse can happen either locally, say in the breast, in the case of breast cancer, or at distant sites um, to form a metastatic recurrent tumor. And this is clinically problematic because the metastatic recurrent tumors, especially, are frequently resistant to therapies, not always, but often resistant to the therapies to which the antecedent primary tumor was sensitive. So again, I don't need to tell this audience the importance of breast cancer recurrence, but it's responsible for the majority of deaths from breast cancer, due in part to this therapy resistance. And recurrent breast cancer and really all recurrent tumors are thought to arise from this population of residual cancer cells that survive following a potentially curative therapy. I want to make this point that sort of I guess an overarching hypothesis of my laboratory and I think a number of labs is that these residual cancer cells or minimal residual disease in patients is analogous to a phenomenon or a type of cell that we call drug-tolerant persister cells that have really been identified and studied in vitro experimental contexts in the laboratory. And I think that that's still a hypothesis or still a thesis. I don't think that that's been definitively proven, but we certainly think that these drug-tolerant persister cells that we are able to study in the lab probably share a number of both cellular properties, but also sort of molecular pathways underlying their behavior with the residual tumors that are present in people. So why do we care about these? We know that from clinical data that patients that achieve a pathologic complete response, for instance, in a response to neoadjuvant chemotherapy, have a much better outcome than those that don't. And this is true not just stratifying patients into PCR, non-PCR, but also quantifying residual cancer using the residual cancer burden index, where patients with a high residual cancer burden, RCB3, have a particularly poor prognosis, which just is, I think, sort of formal proof of what's an intuitive concept, which is both the presence and existence of residual cancer in the breast following a neoadjuvant therapy is associated with increased risk of recurrence. So in this case, by biopsying bone marrow and enumerating the frequency of disseminated tumor cells, it's a number of studies, including meta-analyses, have shown that the presence of DTCs is a poor prognostic factor. So patients who have DTCs in general do worse than ones who don't. But importantly, related to sort of the persistence of residual disease, if patients remain DTC positive for a year after therapy, so these are the purple patients, they have a particularly poor prognosis. So it's again just kind of providing more formal proof of an intuitive concept, which is that the persistence of DTCs or residual disease at distant sites is associated with poor prognosis. So my laboratory's kind of overarching goal is to understand, just like there's hallmarks of cancer with respect to sort of primary cancer cells, we think that these residual tumor cells or these persister cells have their own hallmarks of cancer, which in some cases are similar to, but in many cases will be distinct from the hallmarks of a primary tumor cell. And if we can understand the biological properties of these cells, we think that that will aid in our efforts to eradicate them and ultimately prevent or at least forestall tumor recurrence. That's the goal of my lab. And so I'm going to tell you about some of the models that we're using and some of the specific pathways that we've gotten interested in. But I thought I would just use this slide as an opportunity to have a sort of a big picture discussion of where our work falls. Much of the work we're doing is focused on the cancer cell metabolism of these persister cells or these residual cancer cells. I'm going to tell you a story about a protein called NRF2 and how it regulates both the energy balance in the cell and the antioxidant pathways in the cell to promote their survival. We are interested in genome instability. We're interested in the immune microenvironment and how changes in the immune microenvironment of residual tumors, both locally and at distant sites, influences their behavior. And finally, we're interested in sort of broadly in non-genetic mechanisms of tumor recurrence as well. Okay, but I'm going to really talk primarily about cancer cell metabolism. So why cancer metabolism? Why do we think that it's interesting and informative to study the metabolism of these residual tumor cells? Well, and what are the specific sort of questions that we're interested in? Well, the first is focused on sort of the responses of a tumor to treatment. And so, how understanding how cancer cell metabolism changes in response to anti-cancer therapy and how these metabolic changes contribute to the efficacy of anti-cancer therapies. And then sort of the second part of this is thinking about the relapse and the waking back up of these dormant residual cancer cells. How do these drug-resistant cancers rewire their metabolic pathways? And is this rewiring associated with what we call in the lab new therapeutic vulnerabilities? Um, okay, so those are the questions, but now to the question I teased earlier. Why do we think that metabolism is a relevant pathway to study? Well, sort of the fundamental reason is that work over the past probably 25 years or so has established that oncogenic signaling in oncogenes, so in breast cancer, that would be things like HER2 and MYC and PI3 kinase, which is mutated frequently, but also probably non-oncogenes like the estrogen receptor. So proteins that directly drive the proliferation of primary tumors do so in part by directly rewiring cell metabolism. So this is not just a secondary consequence of sort of unrestrained growth, but instead there's a direct relationship, for instance, between the PI3 kinase AKT pathway and things like protein synthesis and nucleotide synthesis. There's a direct relationship between the MYC transcription factor and genes in the glycolytic pathway, and it goes on and on. And so there's really a very intimate relationship between oncogenic signaling and cellular metabolism. And so then the question and those sort of consequences are laid out on the right here. It really impacts all metabolic pathways in the cell. A second reason is that there's a relationship between the proliferative state of a cell and their metabolic state. And so this is an example from T cells, for instance. But the same is true in cancer, where when you induce a cell to proliferate, it really rewires its metabolism in a way that supports that cancer cell proliferation. And then the final reason we think that this is interesting is that a number of cytotoxic chemotherapies actually directly target intermediate metabolism. And so the most sort of common are things like antifolates that target dihydrofolate reductase and things like 5-FU or capecitabine, which is used in breast cancer, of course, that targets the metallate synthase. And gemcitabine, which is used in pancreas cancer, among other types. And so for all of these reasons, we think that really understanding how metabolism and changes in metabolism are related to these persister cells and changes in proliferation and oncogenic signaling is really important. And so how do we actually study this? And so we primarily use mouse models and in vitro systems to address these questions in the lab. And the mouse model that we use as sort of our workhorse is shown here. And so I'll just walk through this briefly. And so the way that this works is we are able to make genetically engineered mice that express an oncogene, in this case, the oncogene HER2, which is, of course, amplified in about 20% of breast cancers and is the defining feature of HER2 positive breast cancer. We're able to create mice where we can express HER2 in the mouse mammary gland under control of the antibiotic doxycycline. And so when we do this, mice get mammary tumors, and that's shown in this mouse here. And these tumors histologically look like a sort of invasive adenocarcinoma that you would see in people. They express high levels of HER2. At the gene expression level, they resemble HER2-enriched breast cancers in women. But the feature of these that makes them suitable for studying residual disease is that if we now have a mouse that has a HER2-driven primary tumor, and we remove doxycycline, the HER2 gets shut off. So HER2 signaling gets shut off, and the tumor undergoes this dramatic regression. And so this, we think, in some ways, is analogous to treating a HER2 amplified patient with a HER2 inhibitor. Now, it's probably more analogous to an RTK inhibitor like neratinib or lapatinib than the monoclonal antibodies like Herceptin that are used more common clinically. But nonetheless, we think that this is a sort of a genetic model for an anti-HER2 targeted therapy. So the tumors regress and the mice are ostensibly tumor-free. So we would say that they're, in either cured or in remission. However, if we just wait and we monitor these mice over a long period of time, ultimately the mice develop recurrent tumors, similar to a patient developing a relapse following anti-HER2 targeted therapy. So on the left here is shown the tumor growth curves both um, for mice with this in this model, and you can see that they develop these HER2-driven primary tumors, and then following DOX withdrawal to turn off HER2, you get this really rapid and profound tumor regression, followed by this minimal residual disease state, where we know there must be cancer cells in the mouse's body, but they're not macroscopically visible. And then ultimately, these mice develop relapses somewhat stochastically. We like this model because, at least relative to the lifespan of the mouse, these recurrences happen both stochastically but over a long period of time. And so on the bottom left is a Kaplan-Meier recurrence-free survival graph of these mice. And you can see that the median recurrence is approximately 200 days, which is a significant fraction of the mouse's life. So analogous to patients with breast cancer, where recurrence happens over a sort of a protracted period, these recurrences happen over a significant duration of the mouse's life. So the reason these models are useful is that we know that the majority of these mice are going to recur. And so we know that the majority of these mice, the vast majority of these mice, must have residual cancer cells in their mammary gland, which is the mouse analog of the human breast. And so what we're able to do is to sacrifice a mouse that has a primary tumor and look at the mammary gland and see if we can find and characterize the residual cancer cells. And so on the bottom left here is a whole mount of mouse mammary gland that's normal. So this mouse never had a tumor. This dark structure here that I'm pointing to is the lymph node. So that's a normal structure. And then the purple is the ductal epithelial tree. On the top here is a mouse that previously had a tumor that is ostensibly tumor-free, but you can see that in this whole mount, you see the presence of an unusual scar-like lesion here. And when you put this gland into paraffin and section through it, you can see the lymph node in the middle here, but you can see the presence of these multiple foci of residual cancer, one of which is blown up here. Then we can go and characterize in various ways. And so we can characterize these biochemically or genetically or histologically. And so just as an example here, we've shown Ki67 staining indicating that the residual tumor cells that are present here are non-proliferative. They're dormant as compared to a primary tumor, which is rapidly growing. Okay, so using this model, we first wanted to just ask the question, how does cellular metabolism change in response to sort of oncogene inhibition in response to an anti-HER2 therapy? And then how does it change in these residual cancer cells? And so the first thing we did was to do FDG PET imaging of the primary tumor. And so you can see this is the primary tumor here, or this primary tumor very shortly after we've extinguished HER2 signaling. And what you can see is that the tumor very rapidly becomes FDG negative, so it loses its FDG uptake. Even prior to any changes in tumor regression. So there's still a tumor here. The tumor is actually the same size as here, but you get this really rapid loss of glucose uptake. And that's quantified in these multiple models where both tumors are driven by HER2, but also the oncogene MYC, which is frequently amplified in triple negative breast cancer, and the oncogene Wnt1 as well. So this loss of glucose uptake seems to be a common feature of oncogene inhibition in diverse breast cancer models. So what are the consequences of this for the cell's metabolism? Well, there's a protein called AMPK, which is an energy sensor that senses the ratio of ATP to AMP, and it generally responds to bioenergetic stress in the cell. And when it's phosphorylated, the cell is experiencing bioenergetic stress, and it sort of shuts down anabolic metabolism. And what you can see here is that AMPK phosphorylation goes up rapidly following, in this case, HER2 extinguishing, but ultimately gets resolved by the time these recurrent tumors form. And so this suggests to us that cells are experiencing metabolic stress as a consequence of oncogene inhibition, but that metabolic stress has to ultimately be resolved in order for the tumors to recur. Okay, so, and I should have said this, this is work that was done by a former graduate student in the lab, Doug Fox, who's now a postdoc fellow with Dan Haber at MGH. Okay, so at the sort of the 20,000 foot view, cells are losing glucose uptake and they're experiencing bioenergetic stress. But we want to get a finer view of the metabolic changes that were a consequence of HER2 extinguishing. And so we turned to an in vitro mammosphere model that kind of mimics a lot of the stages that I described in the in vivo model. And so let me just describe briefly how this works. And so on the left here is a mammosphere that's derived from a mouse tumor from the mouse model that I described a couple slides ago. And so you can see that these cells form kind of a nice tightly packed sphere of epithelial cancer cells that are proliferative. And so the Ki67 staining is shown in green. And in general, they're not undergoing any apoptosis. So cleaved caspase 3, which is a marker of cell death, is shown in red. And so these are proliferative and sort of happy primary tumor cells that are driven by HER2 because we put doxycycline in the cell culture media here to keep HER2 on. And then what this system allows us to do is, in a very clean way, ask what the consequences of turning HER2 off are in a system where there's only cancer cells around and there's none of the host cells. And what you can see is when we turn HER2 off, the sphere sort of starts to become a little bit disaggregated. Proliferation stops. You lose this Ki67 positivity. And then about a week later, you get this uptick in apoptosis. And ultimately, about 2 weeks later, you get a loosely aggregated cluster of cells that can actually remain in a viable but non-proliferative state in culture for several months. And we've shown this in experiments that I'm not showing here. And so we think that this is analogous to sort of primary tumor growth, tumor regression, and residual disease, similar to what we see in the mice. And so the question that we wanted to ask is, can we use this model to uncover metabolic changes that are associated with HER2 inhibition in this 3D mammosphere model? So to do this, we did liquid chromatography and mass spectrometry based metabolomics. This is just a heat map showing the metabolites that change when you turn off HER2. There's a significant number of them. But the ones that we were most that were most striking, that we were most interested in, were metabolites that reflected a loss of redox homeostasis. So and indicative of the cells experiencing redox stress. And so a couple of those are shown here, where the ratio of oxidized to reduced glutathione and the abundance of oxidized NADP plus and a reduction in the levels of reduced NADPH are all observed following HER2 extinguishing. So this suggested to us that these cells, these cancer cells, when you turn off HER2, were undergoing oxidative stress and had an increase in reactive oxygen species. And so to measure that directly, we use this dye called DCFDA, which measures reactive oxygen species. And you can see that these mammospheres are becoming very, very strongly positive for this ROS specific dye. Interestingly, we wanted to see if this was a general phenomenon of targeted therapy in breast cancer. And so we used a panel now of human breast cancer cell lines shown on the top here, treated with targeted therapies against the cognate oncogenes present in those. So just as an example, BT474 cells and MCF7 and T47D cells have PI3 kinase mutations that partly drive their growth. And you can see that the PI3 kinase inhibitor BKM120 induces ROS, indicated by these orange squares here, in at least the T47Ds and BT474 cells. And then the same thing, a HER2 inhibitor, lapatinib or neratinib, induces reactive oxygen species in the HER2-driven breast cancer cells, BT474 and SKBR3. So we think a loss of redox homeostasis is a common phenomenon following loss of oncogenic signaling in breast cancer. And so what are the consequences of this? What's the functional significance of this? Well, to try to answer that, we wanted to see if we could rescue the increase in reactive oxygen species, in this case using a couple different antioxidants, N-acetylcysteine, or we've done this with glutathione here on the right as well. And now what I'm showing here is human breast cancer cell lines treated with the HER2 inhibitor lapatinib, where you can see there's a loss of viability that's rescued almost completely, interestingly, by these antioxidants, suggesting that the loss of redox homeostasis is contributing in a pretty significant way to the cell death induced by these receptor tyrosine kinase inhibitors. Okay, so our model thus far is that we have a primary tumor which is proliferative, and either therapy like a RTK inhibitor or a PI3 kinase inhibitor or a genetic model of anti-HER2 therapy induces oxidative stress, and that this is contributing to cell death. But we know that there's a population of cells that are able to survive oncogene ablation or oncogene inhibition and remain as these dormant persister cells or residual disease. And so what this suggested to us is that there must be endogenous pathways that are able to counteract this oxidative stress. So in our experimental system, we supplied these antioxidants, these chemical antioxidants, but we think that there's probably endogenous antioxidant pathways that are able to help cells, at least a fraction of cells, survive the loss of oncogenic signaling. And so as we started to think about what these might be, our attention was drawn to a protein called NRF2, which I'll call NRF2, which is a transcription factor that's really the cell's main tool, or the main pathway that cells use to deal with and mitigate oxidative stress. And so the way this protein works is it's actually the gene is constitutively transcribed. Under normal basal or homeostatic conditions, it's very rapidly degraded through the function of this KEAP1 and COL3 ubiquitin ligase complex. However, upon oxidative stress or other stresses, including metabolic stress and electrophilic stress, there are some KEAP1 undergoes oxidation of some key cysteine residues that leads to NRF2 dissociating from KEAP1, ultimately accumulating in the nucleus and driving the expression of its target genes, which are involved in mitigating oxidative stress. So these are things that are involved in making glutathione and making NADPH, among other things. Interestingly, KEAP1 is both KEAP1 and NRF2 are commonly mutated, genetically mutated in lung cancer and some other tumor types, but not in breast cancer. And I'll come back to that in a few slides. So this is just a sort of some of the suite of target genes that NRF2 regulates. But the punchline is that you really need to know is at the bottom here, which is that NRF2, when it becomes activated, helps to mitigate reactive oxygen species to restore a cell to redox homeostasis. So we wanted to ask, following HER2 downregulation, do we see evidence for NRF2 upregulation, and what functionally is NRF2 doing in this process? And so we first looked at tumors shortly after we turned off HER2, and this is a Western blot on the top and immunofluorescence staining on the bottom for NRF2, and you can see that NRF2 gets stabilized at 2 days following doxycycline withdrawal. Remember, this is a time point where the cells are losing this glucose uptake. The NRF2 target genes, or at least a subset of them, are also upregulated. Then when we look at these residual tumors, so this is a month, I think, following turning off HER2, you can see that now almost every cell in the residual tumor has upregulated NRF2 here and has stabilized the protein, suggesting that this endogenous antioxidant response is really a key driver of these residual cells' ability to survive. So we expected then, if we were to look at recurrent tumors, that what we would see is similar to what I showed you a couple of slides ago with that AMPK metabolic stress sensor, where we assumed that by the time recurrent tumors formed, that metabolic stress will have been resolved and NRF2 levels would have gone back down. But that's actually not what we found. We actually found that both the expression of NRF2 itself as well as NRF2 target genes remains high in recurrent tumors, suggesting that NRF2 is getting turned on sort of through an adaptive mechanism in response to loss of redox homeostasis, but that once turned on, it's actually getting sort of stuck on, is how I like to phrase it. And these recurrent tumors, even after they've resolved their metabolic stress, still have high levels of NRF2, and that'll become important as we're thinking about ways to target NRF2. Okay, so now what I've shown you is that this oxidative stress or loss of redox homeostasis that occurs following therapy leads to an adaptive NRF2 response that we think then goes and mitigates oxidative stress. And so the question is then, what effect does this have on tumor recurrence? And so to address this, we took a genetic approach where we expressed a mutant of NRF2 that does not bind to KEAP1, so it's not subject to KEAP1 negative regulation, so it's a constitutively active mutant. And we genetically manipulated tumor cells to either be control or express this mutant of NRF2. And we put these cells back into mice. And as you can see from this Kaplan-Meier, they grow tumors at the exact same rate as control tumors. So that's great. And then we removed doxycycline to turn off HER2. So now the tumors regress. And then we just monitored these mice and asked what happens to their recurrence? What are the kinetics with which they develop recurrent tumors? With the hypothesis being that the ones that express a constitutive active NRF2 should recur more quickly. And that's exactly what we saw, as shown in this recurrence-free survival plot on the bottom right here, that constitutive activation of NRF2 drives recurrence. We found the converse was also true, that if we now use genetic approaches to reduce NRF2 levels in recurrent tumors, using in this case shRNA, you can see that the tumors grow more slowly when implanted into mice with both two different reagents there. And so this suggests that NRF2 is both sufficient to accelerate recurrence, but also required for the growth of recurrent tumors. And that's schematized here. And so we were curious, what at the cellular and molecular level NRF2 was doing. We suspected it was helping cells mitigate loss of redox homeostasis, but we wanted to confirm that. And so what we did is turn back to metabolomic analysis, and we knocked down NRF2 in recurrent tumors and performed LCMS-based metabolomics, and then looked at the pathways that were altered following NRF2 knockdown. And you can see in the red box here, one of the key pathways that was altered is glutathione metabolism. And this was a nice control. This is sort of what we expected NRF2 to be doing. An example of that is just shown here, that using this ratio of reduced to oxidized glutathione as a measure of sort of the redox health of a cell, when in primary tumors, if you set that ratio to be 1, you can see that when you turn off HER2, the ratio goes down, indicative of an oxidative state. When the tumors recur, they've restored that, but that's NRF2 dependent, because when you knock down NRF2, you lose that redox homeostasis. And you can also see that with the reactive oxygen species dye that I mentioned earlier. Then we went on to show that this was due to NRF2's ability to control some of the key genes that it's known to control, including things involved in glutathione synthesis and protein homeostasis. So this was kind of a nice control, and this tells us that one of the things NRF2 is doing in recurrent tumors is regulating redox homeostasis. But what was more surprising was that we also saw changes in nucleotide metabolism in these cells. And so when you knock down NRF2, you get a loss of pyrimidine, purine, and nucleotide sugars metabolism. That's just an example shown here, where really almost every nucleotide or deoxyribonucleotide in the cell was altered following NRF2 knockdown. We found that that's due to NRF2's ability to control this gene in the pentose phosphate pathway, which ultimately makes this ribose sugar that's required for nucleotide expression. So this was a little bit of an unexpected result, and suggests to us that that NRF2 is really doing two kind of distinct things. It's both regulating the redox homeostasis of these persister cells, but we think that importantly, once the cells start to proliferate, one of the reasons that NRF2 gets sort of stuck on is that it's now been co-opted to drive nucleotide synthesis in these rapidly growing recurrent tumor cells. And so to kind of summarize what I've told you is that, in primary tumors following therapy, you get this loss of redox homeostasis, which is really key to how targeted therapies work. We think that this is, one of the ways that they work is causing a loss of redox homeostasis, and that's really contributing to cell death following therapy. But that also results in this adaptive upregulation of the NRF2 antioxidant pathway, such that residual tumors, at least in this model, all have high NRF2 expression. And that's both promoting the survival of these cells through glutathione and redox homeostasis, but also ultimately driving anabolic metabolism in these recurrent tumors, including nucleotide metabolism. So we were, interested in understanding more about NRF2 in breast cancer, and also about the metabolic pathways that are sort of associated with this metabolic rewiring that happens. And so in the last maybe 10 minutes or so, maybe 7 minutes or however long I have, I'm going to tell you about just two little vignettes that are ongoing that have sort of expanded upon this work. Um, The first thing I'm going to tell you is focused on sort of these recurrent tumors here that have high NRF2. And we really asked a very simple question, which is what are the gene expression programs regulated by NRF2 in these recurrent tumors? So I showed you some of the metabolic pathways using metabolomics, but what about the gene expression program? Since NRF2 is a transcription factor, it seemed a reasonable place to start. So what we did, this is work from a postdoc in the lab, Yasmin. What we did was just do RNA sequencing of a number of breast cancer models with high NRF2 expression, where we knocked down NRF2, as is shown here, and then looked at gene expression changes. And what we found is as again, kind of a nice control, that things involved in xenobiotic pathway metabolism and the reactive oxygen species go down when you knock down NRF2. So that tells you NRF2 is doing what we think it should be doing. But what was very surprising, and I think really exciting for us, was that we actually saw a number of pathways go up pretty profoundly when we knocked down NRF2. And these were things involved in sort of a pro-inflammatory signaling. So things like an interferon alpha and interferon gamma gene expression response. So a couple examples of that are shown here. So on the left here is a bunch of interferon inducible genes. And what you can see is when you knock down NRF2, the expression of all of these genes go up. In the middle here is a number of transcription factors that control the interferon sort of pathway, including IRF7, IRF9, and STAT1 and STAT2. You can see that all these transcription factors go up when you knock down NRF2. And on the right are a number of cytokines that function to recruit different immune cells. Many of these are downstream of interferons. You can see that they also go up. And so this was suggesting to us that NRF2, in addition to regulating the cell intrinsic pathways I told you about, might also be modulating cancer cells and maybe residual cancer cells interaction with the immune microenvironment. And so we wanted to broaden the models that we were using. And so we looked at a number of additional, these are mouse tumor models that have high NRF2, and we kind of are focused on EMT6s and 66CL4 tumors. And we did the same experiment where we just really simply knocked down NRF2. And what we found is that, as expected, the NRF2 target genes go down when you do this. So NRF2 is functioning as expected. But interestingly, this also leads to upregulation of the interferon-related pathways. And so these are the same interferon-inducible genes I showed before, where you can see that they're going up. The transcription factors like IRF7 and IRF3 and the STAT1 and 2 transcription factors are also going up in these cell lines when you knock down NRF2. So we think that NRF2 is broadly and generally suppressing inflammatory signaling in breast cancer. So these are all mouse models, which we're interested in using for reasons I'll talk about in a little bit. But what about human breast cancer cells? Well, what we did was mine the cancer cell line encyclopedia data to identify cell lines that we predict, breast cancer cell lines that we predict should have high NRF2 levels, and some of those are shown in red here. And we confirmed, at least in 2 of the cases, that they do in fact have high NRF2 levels, shown here. And then in green are a number of cell lines that are controls that have low NRF2. And then we took the same approach, which is to knock down NRF2 in these human breast cancer cells, and then the knockdown is shown here. And then we did gene expression profiling, and the results you can see here were the same, which is that we get this induction of an interferon alpha type response in these breast cancer cells. And so on the basis of this, we think that NRF2 might be affecting how cancer cells interact with the tumor immune microenvironment through induction of this inflammatory response and upregulation of some of these cytokines. And so we went back to our mouse model, and the advantage of the mouse model here is that we can implant these tumor cells into mice with that are fully immune competent with a fully intact immune system. So we can understand how the cancer cells with and without NRF2 interact with the host immune microenvironment. And so we've done this in several different ways. We took our recurrent tumor cell model that I told you about at the beginning of the talk and implanted them into syngeneic FEB mice. And you can see that the tumors grow more slowly. We took our EMT6 cells with NRF2 knockdown and implanted them into syngeneic BALB/C mice, and you can see the tumors grow more slowly. And then the same thing with these 66CL4 cells with NRF2 knockdown and implanted them into syngeneic BALB/C mice. So in all of these cases, NRF2 knockdown slows the growth of these tumor cells in an immunocompetent setting. So is this associated with the change in the immune microenvironment? We found that it is. So as you can see here, this is immunohistochemistry staining for a T cell marker, CD3. And you can see that when you knock down the NRF2, you get a pretty dramatic dramatic increase in CD3 cells. Um, the same is true for CD45 cells, as shown here, and the same is true of NK cells. So I think maybe you can only appreciate that in the graph here as well. So we think that NRF2 is activation is in general suppressing the immune system or the immune response to breast cancer. So we wanted to see if this was true in had evidence of this in human, actual real human tumors. So what we did was we mined TCGA data, and what we did was using RNA sequencing, we divided tumors into those that have predicted NRF2 high pathway or low pathway. And then we used an algorithm called CyberSort to infer or predict the immune microenvironment of those tumors, again, based upon RNA sequencing data. And what you can see is that for CD8 cells, CD4 cells, and NK cells, CyberSort predicts that human breast cancers with high NRF2 pathway activation will have lower abundance of each of these immune cell types than tumors with low NRF2, suggesting that there is evidence in human tumors, again, that NRF2 is an immunosuppressive. And so what I've told you is that in addition to sort of regulating glutathione synthesis and regulating nucleotide metabolism, that we think a sort of a third thing that NRF2 is doing is leading to suppression of the immune microenvironment, and that is contributing maybe to the growth of these, to the regrowth of these either dormant cells or recurrent cells. So we're, currently trying to understand mechanistically a number of questions about this. One is, how does NRF2 activation suppress inflammatory signaling? What's the mechanistic basis of this? And then we're collaborating with Chris Lee to try to understand, using his Bravo cohort, what the relationship is between NRF2 activation, the tumor immune microenvironment, and likelihood of recurrence in human breast cancer. Turns out that there's, the reagents or the antibodies to detect NRF2 at the protein level in human samples are not great, and so we've been working really hard to try to optimize that before we get to the staining on Chris's Bravo tumors. But that's something that we're certainly interested in doing. And then I think most importantly, an implication of this work is that that if one of the things that NRF2 is doing in breast cancer is generally creating an immune sort of suppressive microenvironment, that would suggest that in approaches to inhibit NRF2 may sensitize tumors to immune checkpoint inhibitors. Now, when I was starting to work on this pathway, maybe 5 or 6 years ago, I would always say, but NRF2 is a transcription factor, and it's really probably not druggable. But that's actually changed pretty, I think, in a really meaningful way over the past several years. So there's a number of companies, including Vividian Therapeutics in San Diego, that have developed small molecules that are really effective at both activating, but more importantly for our work, inhibiting NRF2. And they're actually already in clinical trials and several indications. And so I think, one of the things that we're interested in doing is testing whether these approaches would of inhibiting NRF2 would sensitize to checkpoint inhibitors. So we're taking currently, in collaboration with Nancy Davidson, a sort of a pharmacogenetic approach using not the drug from Vividian, but a genetic model to target NRF2 in breast cancer cells and see whether that renders the tumors more sensitive to checkpoint inhibitors. And so that's work that's ongoing and we're very excited about. So I can either stop there and take questions. I think I don't remember if I was supposed to go to 45 or to 12:45 or 12:50, or I can tell this final vignette. I guess I would maybe prefer to stop and take questions, because I think if I do the final vignette, it will go over time. So how does that sound?
We're really okay with whatever you'd prefer, James.
Okay. Yeah. Well, why
I
don't
think
we? Maybe I'll stop and take questions then, because I want to make sure that if there are questions, I'm happy to take them. And the final section actually gets even more into metabolism weeds, so maybe this is a good place to stop and yeah, happy to take any questions. And I'm going to stop sharing.
to take you into the metabolism weeds, When we do give a lot of capecitabine in breast cancer. Actually, it's a very active treatment for most patients, and it's very tolerable. So it's the 5FU pathway is important, and not just GI cancers, but also in breast cancer. But one of the things that's frustrating for me as I think about the applications of these studies is you can give 5FU as a bolus, you can give it as a pump, or you can give it as oral therapy, or you can give it with, I don't know, levamisole and other crazy things weekly that people do with other tumors. Not we don't do that in breast, but in breast, in the HER2 positive patients, we give it with tucatinib, very active and gets across the blood-brain barrier. So really, really important to us. So when you're thinking about these drugs and your models in vivo, in vitro, and in vivo with mice, how do you think the dose schedule compares? I mean, because I think this is a major problem, and obviously, you guys have to go home at some point, so you're not going to feed, your cell line. I mean, I don't know, to do what we do with the patient with like oral therapy or a continuous infusion pump, you need to like put a pellet in there and have it, degrade or something. I don't know. Anyway, how do you think about it?
Yeah, I think it's a great question. And it's something, I'm from maybe I can answer your question partly from um, sort of a basic cell biology mechanistic perspective. So with all of these drugs, and I think 5FU is a great example with whether, administered as this prodrug capecitabine or directly as 5FU, um, the cells actually, the drug actually has to, well, in the case of capecitabine, be metabolized. In the case of 5FU, it has to get into the cell and interact with the cellular pathways. And so I think that the exactly how it does that probably depends a lot upon the dose at which it's given and the time frame over which it's given.
Mhm.
So we've been thinking a lot about these solute carriers that are these proteins that sit on the surface of cells and probably in many cases are required for import of all chemo drugs into a cell. I mean, there's that might not be universally true, um, but as an example, um, anthracyclines like doxorubicin or probably need expression of these nucleoside transporters like SLC28 and 29 to get into cells. And in fact, SNPs in SLC28A3, I think, are associated with cardiotoxicity in patients receiving doxorubicin, suggesting that it's a clinically relevant sort of modulator of drug sensitivity. And so I think if we just give, to your question, if a huge bolus in mice of 5FU that's 1,000 times what a patient would see, the mechanisms by which the tumor is responding are probably quite different than if we give a dose that's more close to what the patients see, and certainly like if we give it over the time frame that the patients are likely to see it. And so that was kind of a long-winded way of saying I completely agree with the premise of your question, which is that that different delivery modes and doses will probably have different biological effects, and that it's important to us to model, like we need to model it correctly. So now, I don't have any good answers about how to do that other than the hard work of actually, measuring the effective concentration that gets to the tumor and having pharmacodynamic measurements of tumor response and so forth. But I agree with the premise of the question for sure.
Really interesting talk. I'm Darian Reed-Brino. I'm one of the breast med oncs and a postdoc in Patrick Patterson's lab. I have a couple of questions. The first one is I'm curious about when in your model with the HER2 where you turn it off, when the tumors recur, have they recovered HER2 signaling, or like is that whole pathway on again and it's behaving like a HER2 amplified tumor, or is it like picking up a second mutation or some gotten around in some way and like the molecular subtype has switched? I'm just curious if you looked at that.
Yeah, it's a great question. They do not turn back on HER2, so they have not just escaped through HER2. In about 40% of cases, they amplify c-Met um, as a sort of a parallel pathway, which probably doesn't happen that much in breast cancer. I mean, you can tell me, but I don't, not unlike lung cancer
Small
where
number.
small number, yeah.
Yeah.
And
They
in triple negative, you find that in triple negative,
Is
yes.
that right? Okay, yeah. And those are so those are sensitive to MET inhibitors for sure, and that so the MET amplified ones are dependent on MET. They've undergone EMT, a full EMT, and that seems to be important for their recurrence. There's other genetic things. They've lost P16 and P19, so the CDK and 2A locus. But there's a good chunk of them where there's really, I mean, they've actually universally lost CDK and 2A. So I think that's a prerequisite for the survival of a cell following HER2 extinguishing. That's not by itself sufficient to drive recurrence. Um, there's a good fraction of them though that really don't have an obvious genetic driver, like another RTK or something. I think at the subtype level, they look a lot like Claudin low, or maybe well, either Claudin or mesenchymal like TNBC, the ones that don't have MET at least. So yeah.
And then in terms of like if you're thinking about the field of the like mammary gland and you had all those areas where you initially had tumors, is it like what proportion of them are coming back with the recurrent
Yeah, that's a really
recurrences?
good question. That's actually hard to answer. Um, we know at the level of the mouse, almost 100% of mice develop recurrent tumors. At each mouse gets 4 to 5 primary tumors, so we don't think that every single gland gets a recurrent tumor. And as you said, in each gland, there's multiple foci. So um, this is an indirect way of answering your question, but we've done a variation of this experiment where we take the tumors out and we put in a DNA barcode and then we put them back into mice. And when we do that, in many cases, the recurrent tumors are derived from a single barcode. So they're truly
One.
clonal. Yeah. We actually do have polyclonal tumors where they're derived from thousands of barcodes. And so you can have sort of both polyclonal and very clonal relapses, and we don't understand at all the difference between those.
And then I have one more. I don't mean to like hog
Yeah.
your time.
Go for it.
So the version of NRF2 that you're using in your model that's constitutively on, if I
Mhm.
understood correctly, it's on from the beginning. Is that
Yeah.
right?
Yeah.
Do you think that's changing the tumors? And then like, and maybe this is what you were getting at with working with Nancy about, but like having it regular and then later making that switch as if,
Mhm.
later in the scheme when it's
Yeah.
dormant
Yeah.
and then how to turn it off again coming in like a treatment.
Yeah, that's a great question. I actually do think that's a good idea, and I think that would be a good way to do the experiment. We do think having it on all the time is changing the tumor in actually a way that's bad for the cells. So if a tumor cell has HER2 and we add in our NRF2, I think the cells are actually experiencing reductive stress where there's not enough because the cell needs some level of reactive oxygen species to grow for signaling and so forth. So we actually those cells, even though they form tumors, like I showed the Kaplan-Meier, the time to tumor formation is about the same. The cells don't like it. So that's the specific answer, but I think your general point is a good one, which is if we had spatiotemporal control over NRF2, that would be much more powerful. So the way we've done this with Nancy, this is a BCRF grant that she has that she's been kind enough to work with me on, is we're using this DTAC system that Behnam Nabed, if anyone knows him,
Mhm.
works with. So we fused NRF2 to FKBP mutant that's able to be regulated by DTAG. So we knock down endogenous NRF2, we overexpress this, and now we have an ability to toggle NRF2 on and off. So that's what we're doing now.
Perfect. I was going to say that's prime for some cool genetic tools.
Yeah, yeah. Okay, thanks, Erin.
Thanks.
Thanks, James. My other question was just that, it's interesting. I think Darian hit on a really important point about how the model, basically changes the flavor of the tumor. But the other, real elephant in the room is everything else in the body and the tumor microenvironment. There's, sure, we could go back to the drug levels and how your liver metabolizes things and how everybody's body is actually different. But I think the tumor microenvironment is probably extremely important in terms of these dormant cells and sort of why a tumor is trophic to bone or liver or whatever. And I know you've done some work in this area. I mean, how can we capitalize on your models to study that?
Yeah, no, I think that's a really important the question. One of the things a weakness of the model is everything I showed you here is recurrence in the local site, which is capturing some of the biology. I mean, I think I can make the case that um, the fact that residual cancer following neoadjuvant therapy in the breast is prognostic for outcomes at distant sites suggests that there's some biology that's being captured there. But the metastatic microenvironment, the immune microenvironment, the extracellular matrix, all of the things is going to be drastically different. And so we do know that this model metastasizes to the lung and the liver, and we can find cancer cells in the brain. They don't form. It takes a while for them. I don't know that they ever actually fully form macromets, but we can definitely find cells there. And so that's something that we're really interested in is what's the relationship between, um, you know, how do tumors that have metastasized to different sites, how do they respond differentially to HER2 inhibition? How's their metabolism different? How's the microenvironment different? Um, so along those lines, we have a real interest with my colleague Kevin Barry in understanding the role of natural killer cells in these residual tumors, based partly in work on that Sarah Hurvitz and Christina Curtis have done, showing that post neoadjuvant chemotherapy and tumors in the breast seem to have elevated levels of natural killer cells, and that's prognostic. The ability of NK cells to sort of come in following therapy is prognostic, and so but we don't know anything about that how that varies by metastatic site. So those are definitely questions we're interested in, and I think these models they're okay for answering some of them. I do think that the mouse models with intact immune systems are yeah, I agree it's an important point, and those are some of the things we're doing to try to get at that.
Thanks. No, this is all it's important. I mean, I think it is important what goes on in the breast, but you know, we can fix that.
Right.
I mean, the real question is what's going on with the sleeper cells elsewhere that have kind of snuck out. But
Yeah.
anyway, great stuff. Thank you so much for sharing what you're up to. Very helpful for us to have that perspective and really excited about the work you're doing.
Thank you. And I just want to say, I
Yeah.
skipped my acknowledgement slide, but I just want to thank Yuri, who's on the call here, who's been a big help getting specimens for us, and also Chris Lee, again, for the work on the Bravo study. So, and thanks for the invitation. This was fun. As I said, kind of basic cancer cell biologist. I really like talking to clinicians, and I'd be happy to chat with any folks for areas of collaboration.
Great.
All right.
Thanks,
Thanks.
James.
Bye.
Thanks, everybody.